The connection with Sirin Mukhtarova is not good. I cannot hear her. I cannot hear her.
Professor, now you hear me?
Now I can hear her. Yes.
Okay. Again, welcome. It's my great pleasure to introduce you, Professor of Cancer Research at the Massachusetts Institute of Technology, Bob Weinberg. A little bit about Bob Weinberg. He studies how cancer spreads, what gives cancer stem cells their unique qualities, and the molecular players involved in the formation of cancer stem cell and metastasis. In 2013, Bob Weinberg was awarded with Breakthrough Prize in Life Science for characterization of human cancer genes, and in 2021, he received the Japan Prize for his contribution to science. Title of the presentation, Epithelial-Mesenchymal Plasticity and Cancer Progression. And now, Professor, you can begin. Thank you.
Thank you very much. Kaili Kesh, добрый вечер. Я рад вас видеть сегодня вечером. I will begin now to discuss the research in my laboratory that concerns the molecular basis, the biochemical basis of invasion and metastasis. I will ask you now whether you can see my screen. Can you see my screen?
Yes, we see your screen.
Okay, perfect. You can see my screen now, yes? Can you see my screen? Hello? Hello?
Yes, yes.
You can see the screen.
Yes, we see your screen. Thank you.
We see you and hear you.
Okay, good. Can you see that screen? Can you see that?
Yes. Yes, we see you, Professor.
Okay, so I will continue. I will begin now, and I thank you, Очень рад вас видеть, to discuss the mechanisms by which human cancers invade and metastasize. And kindly tell me if you cannot see the screen properly. We know that the formation of a human tumor requires multiple successive steps, here depicted in the context of colorectal carcinoma pathogenesis as previously normal tissues become increasingly abnormal, and ultimately they invade and they metastasize. That is, they spread to distant tissues, in this case to the liver. And this multi-step process that operates within the human colon, as I just depicted it, also operates in a wide variety of other types of human cancers, explaining possibly the fact that each of them requires 20, 30, even 40 years to reach the conclusion of creating a primary tumor. Some of this process was elucidated already in 1989 by the group at Johns Hopkins, where they showed that as the histopathological progression proceeds in the context of colon cancer pathogenesis, leading up to a primary tumor, here depicted as a carcinoma, there are a series of somatic mutations that are acquired successively as a previously normal cell acquires a succession, a collection of somatic mutations. Can you hear me okay? Can you hear me okay?
Yes, we hear you.
Realizing this, one can begin to rationalize these multiple oncogenic mutations, all somatic mutations, because they perturb 5 distinct signaling pathways within human cells, depicted here at the top of the slide, which must be perturbed in aggregate in order to experimentally transform a normal human cell into a tumor cell. But these genetic mutations provoke another question. How important are non-genetic programs in determining the path, the trajectory of colorectal carcinoma pathogenesis, the development of a human colon cancer or other kinds of cancers? Here is one suggestion that in fact, non-genetic programs are also very important. In this particular slide, I show the results of work at Stanford University in which researchers created 2 distinct lung cancer lines arising from very closely related but distinct differentiated cells in the lung. And these 2 classes of cells had an identical set of introduced somatic mutations, and yet, as one can see over here, the transcriptional program, the differentiation program of these two cells caused them actually to be quite different, even though they had the identical set of introduced somatic mutations. And this begins to suggest that the differentiation program of the normal cell of origin from which a cancer arises not only influences the normal cells, but continues to influence the biology of all the derived cells that come thereafter. To state it another way, it is not the case that the process of somatic mutation eradicates the influence of the normal differentiation program of a normal cell of origin, which is by necessity non-genetic. It is an epigenetic process. Here now we confront yet another question. How do normal carcinoma cells invade and metastasize? Do they need to acquire additional mutations beyond those that were required previously to form the primary tumor? And we know, by the way, the profound influences of the normal differentiation program. Just to return to that in a moment, if one transforms experimentally human melanocytes or normal human mammary epithelial cells with the identical set of introduced mutations, the transformed melanocytes create large numbers of metastases throughout the body, whereas the transformed mammary epithelial cells with the identical set of introduced genetic mutations don't metastasize at all. And this again underscores the critical role of differentiation programs in governing the tendency, the proclivity to metastasize. If we go back to the process by which a primary carcinoma will invade and metastasize, we need to recognize the fact that the process moving from a primary carcinoma to invading and metastasizing is itself a complex multi-step process involving local invasiveness, entrance into the circulation, translocation, a transport through the circulation, escape from the circulation into the parenchyma of tissue, and the formation of a micrometastasis. And on rare occasion, in a distant tissue, the micrometastasis will form a macroscopic metastasis. This last step being termed colonization and being extremely complex and poorly understood. This series of steps is together termed the invasion metastasis cascade, and itself begs the question, how do cancer cells acquire all the distinct capabilities that are required in aggregate to execute the invasion metastasis cascade? Do they need additional mutations beyond those that were previously sustained to form the primary tumor? Or is there an alternative process operative? And the answer to that question comes from micrographic images like this one here, in which we see transformed human mammary epithelial cells, that is, mammary carcinoma cells, that have been stained with an epithelial marker, in this case for cytokeratin. And here we see the transformed epithelial mammary carcinoma cells. Out here on the outside, these cells are growing within an immunocompromised mouse, and we see the recruited mouse stroma, the nuclei of the mouse stromal cells. But here, provocatively, on the outer edge of this island of carcinoma cells, we see cells that have shut down the epithelial marker cytokeratin and now express instead a mesenchymal marker, which is termed vimentin. And this suggests that these human cells have undergone a profound change in their biological program from an epithelial to a mesenchymal state. One can see this even more clearly here, where here we have a tongue of carcinoma cells that is stained with cytokeratins. Outside are the recruited host cells from the mouse stroma. And here we see a rim, a border of cancer cells that have shut down the pre-existing marker cytokeratin. And here we see yet other cells that now begin, carcinoma cells that now begin to turn on the mesenchymal marker vimentin. Granting this, we come to conclude that these cells have undergone a profound change from an epithelial to a mesenchymal state because they are in direct contact with the stroma from which they receive signals. That is, the cells on the outside in direct contact with the stroma move from an epithelial to a mesenchymal state, and we call that shift an epithelial mesenchymal transition, or simply EMT. Here is one example of how this might occur at the cellular level. On the left, we see a carcinoma cell that is in the epithelial state. Here it's on the edge of the tumor, as I showed you before. Here is a stromal cell that is recruited from the host. The epithelial carcinoma cell releases a signal, interleukin-1, that impinges upon the stromal cell. And the stromal cell signals back to the carcinoma cell by releasing a series of signals that within the carcinoma cell activate a previously silent, latent EMT program. And so these interactions between distinct cell types are critical to the activation of the EMT, which is a complex, multifaceted program whose activation does not require the mutation of genes. It is entirely epigenetic. It is involved, among other things, in wound healing. And here we can see a series of traits that are shifted when cells move from an epithelial to a mesenchymal state. Indeed, there are 300 or 400 genes that go up and down when cells move from the E epithelial to the M mesenchymal state. Here we see an example of an EMT occurring in a normal sea urchin embryo, where we have these gastrulating cells that are moving from the outside, the epithelial state, into the mesenchymal state as they invade into the center of the embryo, where they create the beginnings of the endoderm and the mesoderm. Here we see in a chordate embryo, cells from the neural crest, and as they migrate out, these blue cells here, these light blue cells, they undergo an EMT to enable them to migrate, to move through the embryo. In fact, the EMT occurs in a whole variety of metazoan embryos, and in each case, in these metazoan embryos, the EMT is expressed transiently in various tissues as a critical component of normal morphogenesis. In fact, the EMT in all these cases is induced by transcription factors whose names are given above each of these micrographs. The EMT was invented a very long time ago, probably in the Precambrian, 700 million years ago, and all metazoa express these transcription factors and the EMT program. Here we see in the context of, for example, cancer progression, that we have epithelial carcinoma cells that have lost the critical epithelial marker E-cadherin in this small space down there. And in that small space, suddenly we see the expression of snail, which is an EMT-inducing transcription factor. So we begin to see some causal relationship between the expression of one of these transcription factors, the EMT program, and the acquisition of aggressiveness. I will change now momentarily to an entirely different topic, which will come back to the one we just discussed. In 2004, Muhammad Al-Hajj and Michael Clark demonstrated that if one takes a series of cells from a breast cancer or breast cancer cell line and dissociates them and fractionates them by fluorescence-associated cell sorting using two cell surface markers, one can stratify, one can resolve this population of cells into a minority population and a majority population. And what one sees is that if one takes cells from the minority population, as few as 200 of these are able to seed a new tumor when implanted into an immunocompromised mouse. Conversely, cells of the majority population, 20,000 of them, fail to form a tumor. And this indicates that these two subpopulations within a given tumor, all with the same genetic lesions, reside in different epigenetic states, one having tumor initiating capability and one lacking it. Here we can see some kind of hierarchical organization as occurs in a normal epithelial tissue. And here we can see in another version of what I just showed you, the stem-like cells are shown up here in the FACS analysis, and the non-stem cells are shown down here. And these are two alternative states that the carcinoma cells can reside in. Some years ago in my laboratory, investigators examined whether there's any relationship between the EMT program that we discussed before and the stem cell program. And to do so, they induced either of two transcription factors, snail or twist, as you can see here on top, into these cells and asked the question, what would the consequences be? And when they did so, they discovered that the cells that previously looked very epithelial underwent a change in morphology and looked very much like fibroblasts, like mesenchymal cells, and they migrated from the non-stem cell state to the stem cell state, giving us the first indication that there's an intimate connection between the EMT program and the stem cell program. Here we can see another context in which the EMT plays a critical role. Here we see a cross-section of a normal mammary duct, and the mammary duct, as you may know, is a bilayered epithelium. It has a luminal layer on the inside and a basal layer on the outside, and importantly, the basal cells carry the normal stem cells in the mammary gland. And what one sees here is that an EMT-inducing transcription factor, in this case called SLUG, is expressed normally in this normal mammary duct. Indeed, it's expressed by more than 2 orders of magnitude higher in the basal cells than in the luminal cells. And these are, as I said before, thought to be stem-like cells. In fact, we find EMT-inducing transcription factors in a whole series of epithelial tissues that have double layers, such as the mammary duct, but we don't find them in epithelial tissues that have only single layers. For example, here the EMT transcription factor is not expressed in the crypt of a colon where there are stem cells. There is a different principle operating here to form the stem cells. Can one prove that the stem cell program is critical for the operations of mammary stem cells, and that the EMT program can induce normal mammary cells to become stem cells? One can use there a very interesting biological assay in which one takes the mammary stromal fat pad of a young female mouse, left in situ, removes the beginning, the incipient mammary ductal tree, clears the mammary stromal fat pad, and then implants in the cleared mammary stromal fat pad candidate mammary epithelial stem cells. Here's what the normal mammary gland looks like. Here's what one looks like after one implants mammary epithelial stem cells, creating an essentially identical mammary ductal tree. In fact, if one implants 10,000 normal mammary cells into a mammary ductal tree, into a cleared mammary stromal fat pad, nothing happens. But if one exposes these cells briefly to EMT-inducing transcription factors, shuts them off, and then looks at 6 or 7 weeks later, the cells that were previously exposed transiently, briefly, to an EMT program, as few as 100 of them can actually now form and seed an entire mammary duct. This suggests that there's an increase in the number of mammary stem cells more than 100-fold, and begins to suggest that the connection between the EMT program and the stem cell program operates both in normal epithelial tissues as well as in carcinoma tissues. In other words, this is the major conclusion. In fact, the EMT program is a series of steps, involves a series of steps from the epithelial state as a cell moves progressively from the epithelial state toward the mesenchymal state. It does not operate as a binary switch moving from one state to the other. Instead, as the EMT program is activated, cells will lose ecadherin, turn on this cell surface marker CD44, turn in vimentin, shut down P63, and shut down integrin beta 4. So it's a series of progressive steps. And in fact, it is the case that in spontaneously arising human cancer cells that activate the EMT program, they never move from E all the way over to M. They only go part way. And here one sees that if one looks where there are stem cells arising, they're present in the middle. These are partly mesenchymal, partly epithelial cells. You see here in this purple curve, this is where the stem cells reside. And if one pushes cells experimentally too far into the mesenchymal state, then they will lose their stemness. Cells in the middle, 1 in 3,000 of them can activate a stem cell program. Cells way over here, 1 in 750,000 of them can seed a new tumor. And therefore, there is a 250-fold difference in their relative tumor initiating cell frequency, where TIC means tumor initiating cell. Indeed, we believe that at each of the stages of multi-step tumor progression, there are stem cells and there are derived non-stem cells that are formed during each of these multiple steps of tumor progression. As cells acquire more and more somatic mutations, they continue to have stem cells and derived non-stem cells that are more differentiated, where each of these sectors in these circles indicates the acquisition of yet another somatic mutation. Here is a proof that the EMT program is involved causally in the formation of aggressive tumors. Here, there have been very benign mammary carcinoma cells that are implanted in a mouse, and what looks 6 or 8 weeks later for what the tumors behave like. And as you can see here in the edge of the tumor over here on the left, where the white line is, this is a very smooth edge, and these cells are therefore non-invasive. On the other hand, if one takes these cells, exposes them transiently to an EMT for a couple of weeks, and then shuts off the EMT program, 6 or 8 weeks later, 10 weeks later, the edges of these tumors are then highly invasive, as you can see here. And this proves directly that, in fact, the EMT program can confer on carcinoma cells an aggressive phenotype. Indeed, if one looks for the presence of metastatic growths, metastases in the lungs of tumor-bearing mice, the parental cells indicated over here form virtually no metastases. But if these cells have been exposed for a short period of time to these two EMT-inducing transcription factors, now one sees 100 macroscopic metastases in the lung and 400 microscopic metastases in the lungs, which I illustrated briefly before. Hence, the EMT program can drive cells to high states of malignancy. And this leads us now to the insight that the EMT program can solve the problem of how physically cells can leave a primary tumor, complete many of the steps in the invasion metastasis cascade, up to the point of forming a micrometastasis, a microscopic growth in a distant tissue. And therefore, this complex multi-step process can be accomplished by this non-genetic program without the need to acquire additional mutations beyond the mutations that were required previously to form a primary tumor. However, now one confronts the last step, and it is my belief that the EMT program cannot influence this last step, cannot facilitate this last step, simply because when micrometastases are seeded into distant tissues, they must undergo complex adaptations that enable, for example, a mammary cell to thrive in the context of the lungs or the brain or the bone marrow. And this is a very complex process which we still do not understand and actually occurs very inefficiently. This therefore represents the major unsolved problem of cancer. We've known for many, many years that different kinds of cancers form metastases with different tendencies in the patient. For example, colon cancers form many liver metastases. Prostate cancers form many bone marrow metastases. Breast cancers form many bone marrow metastases and smaller numbers of lung metastases. And it is these adaptive steps, as you see in the top left, the process of colonization, that somehow must explain the ease or the difficulty that differentiated metastasizing cancer cells are able to form tumors in different sites. And therefore I say, Rahmet sizde, Большое спасибо. Thanks. Мне было очень приятно вас видеть сегодня вечером. And I would be pleased to take questions now. По-английски. Пожалуйста.
Can you hear me?
Now I can hear you.
Okay. Thank you for a great presentation. And we have some questions. We want to ask which factors activate this mechanism of AMT? What provoked AMT program?
There are signals that are released by the stromal cells, signals such as TGF-beta, canonical WNTs, even interleukin-1, which then impinge on the carcinoma cells and induce the carcinoma cells to activate their previously silent EMT program. So we know some of the heterotypic signals that are critical for the activation of this program in the carcinoma cells, this program which is otherwise not expressed.
And another question, can epithelial AMT program will be reversible? Can we stop, interfere this process?
Yes, so the EMT program is highly reversible. The reverse of the EMT program is called the MET, the mesenchymal epithelial transition. And we know that after carcinoma cells begin to initiate a metastatic colony, many of them will then thereafter undergo an MET and become more epithelial again, simply because the more epithelial cells can proliferate more capably than the highly mesenchymal cells. Whether we can shut down the EMT program in human carcinomas through some type of pharmacologic treatment is something that is being intensively explored now in many laboratories. It is still an unsolved problem, i.e., how to shut down an existing EMT program. Because in addition to what I said, cells that have undergone an EMT are actually more resistant to many kinds of therapy. And as a consequence, they are the survivors that create the residue of surviving cells after the bulk of tumor cells have been eliminated by radiation or by chemotherapy or even by immunotherapy.
Thank you. And the last question, is there some difference between pediatric cancer and adult cancer in the mechanism of this AMT or stem cell cancer?
That is a very interesting and provocative question that I often think about, simply because it is clear that the creation of pediatric cancers operates on far different principles than does the creation of adult tumors. It's almost certainly the case that pediatric cancers do not require 3, 4, or 5 somatic mutations, as I showed you earlier in the context of colon cancer pathogenesis. There is a time early in development when the pediatric cells, the cells in the embryo or the newborn, are relatively easy to transform with a relatively small number of acquired somatic mutations. But there's a point soon thereafter when these cells become like adult cells and are no longer so easily transformed. This is a major mystery in contemporary cancer research whose resolution is still unclear.
Okay, thank you, Professor, for your presentation, for the answers.
Спасибо большое. Мне было очень приятно вас видеть сегодня вечером.
Your Russian as well.
Я еще не забыл все моего русского языка.
Thank you very much.
Пока.